My name is Mallory Choudoir. I am an assistant professor and soil microbiome extension specialist in the Department of Plant and Microbiology. Today, I'm here to talk about strategies for cultivating a healthy microbiome. So today I will cover general topics, an overview of the beneficial functions of agroecosystem microbiomes, strategies for rebuilding soil microbiomes following environmental disturbance, and then to uh, finalize, I'll talk about specific things to consider and best practices for using microbial biostimulants to improve soil health or to enhance crop productivity. Okay, so when I say microbiome, I am referring to the collection of diverse microbes, including fungi, bacteria, and viruses, and their genetic material, so their DNA, their genes, specifically within a given environment. So as humans, we have unique microbiomes on our skin, in our guts, and agroecosystems also have unique interacting microbiomes. Soil microbiomes are some of the most biologically complex microbiomes, but certainly there are different microbiomes that are living in the roots of plants, in the tissues of plants, and on the leaves of plants. And these microbiomes carry out critical ecosystem functions for maintaining soil health and supporting crop productivity. In the soil, these activities include soil organic matter decomposition and nutrient cycling. Soil microbiomes govern below ground global carbon cycles for carbon, nitrogen, and phosphorus. Soil microbes enhance water and nutrient uptake by plants, and then directly and indirectly also contribute to pest and pathogen control. And also on broader scales, soil microbes are governing the flow of carbon and nitrogen between the atmosphere and the ground through greenhouse gas emissions. So to sum these beneficial functions up, microbes are global nutrient cyclers and controlling the flux of gas between the soil and the air. Importantly, contribute to soil structure and water retention, and then also plant growth promotion. So one function of soil microbiomes that I think is not often appreciated is how soil microbes contribute to the formation and the stability of soil aggregates. So living and dead microbes adhere to soil particles and release extra, they release sticky compounds that literally stick the soil particles together, contributing to water retention. So here are some images of microbes adhering to soil particles and creating soil aggregates. In relation to plants, soil microbes are also incredibly important for directly and indirectly promoting plant growth. Some of these activities include expanding the genetic and ecological capacity of plants. So with this image here, we have this tomato plant, and these are the billions of cells that this tomato plant will interact with throughout its life, essentially expanding the ecological capacity of a plant. Microbes improve nutrient and water use efficiency and access. Microbes also directly produce plant hormones that contribute to plant development and stress tolerance. So microbes are responsible for contributing to plants' ability to tolerate heat, drought, extreme temperature, stress, and also disease suppression. One particular group of microbes that are incredibly important for plants are mycorrhizal fungi. This group of fungi associate with over 90% of plants. There are two types of mycorrhizal fungi: ectomycorrhizal fungi, which form mycelia on the outside of roots. These are dominant in forest fungal biology, so associating with trees. And then our buskrill mycorrhizal fungi, which are more common in our cropping systems. These relationships are extremely old, have evolved with plants throughout the course of evolutionary history, and over 80% of land plants have associations with AMF. However, not brassica and cabbage, so something to consider when thinking about microbial relationships with our food production crops. And mycorrhizal fungi are important for exchanging nutrients with plants. So here we have an illustration of what the plant is providing the fungus, which is carbon. And in exchange for that carbon, mycorrhizal fungi transport phosphorus and nitrogen into the plant, directly improving nutrient use and access. Okay, so that was a very brief overview of the beneficial functions of microbiomes. And now we're going to transition to strategies for rebuilding soil microbiomes following disturbances. So when we think of what is happening after an environmental disturbance, there are a few ecological scenarios that are playing out. One could be there is no disturbance, so this the microbiome structure stays the same, and also the function stays the same. In this instance, we have the opportunity to maintain or enhance our microbial communities. If this environmental disturbance has somehow altered the community structure, microbiomes are incredibly resilient. And what can happen is, following a disturbance, that same community can recover, also recovering the function, right? So in this case, in addition to maintaining and enhancing beneficial communities, we can also support the recovery of microbes. Sometimes, after a disturbance, a microbial community structure remains altered, but due to the vast activities of microbes, called functional redundancy, the structure remains altered, but the function can recover, right? So in this case, we can also support community recover while maintaining or enhancing. And then this bottom one is if the community structure remains altered and we also do not regain function. These are the scenarios that we are not looking for, right? So we want to do things to support recovery and maintain and enhance activity. So a few things to consider when we're talking about environmental disturbance, right? So different disturbances will have different impacts on the microbiome structure, composition, and function. So important things to consider in the event of flooding or fire, or even fumigation or anaerobic soil disinfestation, right? So all of these impacts are altering soil microbiomes. So are we looking at massive soil erosion or topsoil loss, which might result in low nutrients? Are we talking about sedimentation, which could be a massive change in soil texture, nutrients, potentially contamination, a loss of biodiversity, right, through flooding, fire, or again, sort of imposed fumigation strategies. And when we are thinking about recovery, the first thing we want to think of is, where are the microbes coming from? What microbes are still present? What regional sources of diversity are they sourcing from throughout this recovery? And the strategies we can practice to help microbiomes recover really include the USDA NRCS principles to manage soil health. And I'm going to talk about these with a bit more focus on microbial activity. So one strategy is to feed the existing microbes in the soil with carbon-rich amendments to add bulk nutrients, and this can be through the addition of mulch, compost, and biochar. Maintain living roots through cover cropping or diverse cropping rotations, and minimizing disturbance. Number 2 and 3 are interrelated because cover cropping also minimizes disturbance and decreases soil loss. And then finally, we can also think of adding biology. So adding new microbes, new beneficial microbes to our soil systems through the input of biostimulants. When we think of feeding our existing microbes, we want to be adding organic or carbon-rich amendments. And there are a number of amendments that are available. On the left is biochar. And then on the right are a number of other amendments, including manure, compost, green manure, waste. All of these are viable options. Again, unique things to consider when thinking about adding organic amendments. Certainly the cost and the quality, the potential safety considerations for food and forage. And when we think about quality, we want to think about the ratio of carbon to nitrogen. In general, the ideal microbial diet is a C to N ratio of 24 to 1. Most composts are a bit higher C to N ratio, but the ideal diet for a microbe is 24 carbon to 1 nitrogen. And depending on the material we are adding, in this table, we have a number of different materials in the C to N ratio. We can adjust this a bit by adding different high nitrogen, known as green composting inputs, such as grass or clippings. And that adds in nitrogen if you have a lot of high carbon materials. Conversely, if you need to add carbon to the system, we can add things more brown composting inputs, so wood chips, leaves, straw, and this can be an iterative approach, but we want to target that 24 to 1 C to N ratio. So I want to transition to talking about cover cropping and the important dynamics that are linked between microbes and cover crops. So cover cropping provides living roots for microbes, and this contributes both to soil chemical and physical health. So the roots provide a physical structure and home for microbes, and bacteria are able to excrete extra polymeric saccharides, or EPS. This is that gunky stuff that microbes form aggregates and adhere to soil particles, which improves water retention. The plants provide microbes in the rhizosphere carbon, so improving the plant carbon sink in the soil, which ultimately contributes to carbon sequestration as microbes die and this carbon is protected. And then finally promotes plant growth promotion through the interaction between plant roots and mycorrhizal fungi, as well as other beneficial microbes. And I love this image of the root structures of various cover crops. So, we see the above ground diversity, but below ground, there's so much surface area of cover crops providing unique opportunities for microbes to be recruited and carry out these beneficial activities. So as above, so below, above ground biodiversity will mirror below ground biodiversity. So essentially, each different plant has a diverse buffet of carbon-containing molecules that it will feed microbes in the root system, stabilizing and recruiting diverse soil communities that can then contribute to the next rotation of your farm or production system. Okay, so those are the practices that generally support healthy and functioning soil microbiomes. In this last part, I'm going to transition to best practices for adding biology to your systems. The agriculture biological landscape is a massive commercial landscape in the past few years, and this is certainly growing. So we have so many opportunities to utilize different types of living organisms and other bio-based substances. And while incredibly promising to be harnessing beneficial, naturally occurring microbes, substances, many of these products and solutions are lacking local performance data, and efficacy can vary widely. So while promising, we are still in the phase of identifying strategies that are effective and beneficial. We can think about this landscape of microbial biologicals through their general activities. So biopesticides are naturally occurring substances that control the presence of pests, right? So biocidal activities, and these are highly regulated by the EPA. Conversely, biostimulants are defined as a substance or a microorganism that, when applied to seeds, plants, or the rhizosphere, stimulates natural processes to enhance or benefit nutrient uptake, nutrient efficiency, tolerance to abiotic stress, or crop quality and yield, right? So this is a very general definition. And importantly, there are little regulations to biostimulants. So currently, biostimulant registration is similar to the state level to fertilizers. There is a new biostimulant certification program. However, this is not required. This is an activity, a voluntary certification program, and this group is hoping to improve our ability as consumers to evaluate the use of these biostimulants. However, there are very few biostimulants that are currently certified under this program. And again, this is a voluntary program, not regulated by the state or the government. So just as a quick overview, we have biostimulants, which are either microbes or other products that stimulate natural beneficial processes. These are, for the most part, unregulated, versus biopesticide, which control pests, and these are highly regulated by the EPA. When we think of biostimulants, even there's a broad landscape of what these products actually are made of. Some of these are microbial, so these are living microbes, including mycorrhiza, other plant growth promoting bacteria, including nitrogen-fixing rhizobium. Some of these products are single microbes, some are complex communities. Other products are biologically derived, but themselves are not living microbes. These include humic substances, other organic acids, potentially vitamins, salts, amino acids. And then finally, there are a number of products that are extracts. So seaweed extracts, other plant-derived extracts. So again, these microbial biostimulants can either be living microbes or natural products. So products that are produced by microbes, such as some of these chemicals on the bottom, or once living biological entities such as extractions from seaweed or kelp. So some of these have interchangeable terminologies ranging from biofertilizers, which are biostimulants that improve nutrient use efficiency or nutrient access, ranging to microbial inoculants, which may also be biofertilizers or may provide other beneficial activities for plants. So I have outlined 5 tips, best practices for incorporating biostimulants into your production systems to either improve soil health or crop health or maintain. Number one is to select the product carefully, right? There's a massive landscape of these. Be cautious of silver bullet solutions and big claims. Um, Read the label, right? So understand if this is a living microbe, if it is an extract, what are you putting into your system? Certainly, we need to consider the economic cost and the return on investment. Sometimes this cost can be considered beyond the cost of the product itself, especially if inclusion of a product may result in the need for fewer synthetic fertilizer inputs, irrigation, or other inputs. Does the product target a particular crop or a stress? And then finally, what is your goal? Are you looking to utilize these products for biocontrol or potentially enhance nutrient use efficiency? And then finally, these will work best under optimized growth conditions. Read the label and then reread the label. So on these labels, you will find the guaranteed analysis of what is in this. You can identify if this is a single microbial species. Is it a particular strain? The cell number and density will be on the label. Identify the storage and handling instructions, safety and incompatibility, and importantly, the application instructions. From the label, we can determine or infer the mode of action. Sometimes the MOAs can be general, other times they can be specific. In the case of a biopesticide, this will be a very specific MOA. Other times, this is a general plant growth promoting product. Multiple ecological functions are possible. It is possible to have both biocontrol activity while also promoting other beneficial functions. Another specific mode of action that is common for biostimulants is this product a biological nitrogen fixer, right? So microbes that are fixing nitrogen from the atmosphere and providing that nitrogen to plants. Other groups of microbes are phosphorus solubilizers. This is a known activity, so this would be a product that would help to improve phosphorus utilization and access. Some of these biostimulants are plant endophytes, which would be microbes that colonize a plant and are living inside the root or leaf tissues. Number 4, it is important to follow the storage and handling recommendations. This seems pretty straightforward, but again, it's really important. These are living microbes. Often we want to store these in cool, dry, well-ventilated locations, avoid extreme temperatures, usually less than 80 degrees Fahrenheit and usually above freezing. We want to keep away from direct sunlight. Most of these are single use, so we want to do our best to keep these sealed to prevent contamination. And then finally, mindful of the expiration date. Again, living microbes, past the expiration date, they will likely still be living, but will have reduced viability. I'm a microbiologist. I eat things in my refrigerator that are certainly expired, but always at risk of potential negative outcomes. And then we also want to identify potential incompatibilities and to ensure the correct application. Certain fungicides, herbicides, insecticides, or even other inputs, including amendments or fertilizers, may decrease microbial viability. We want to be mindful of the moisture, the pH. If this is a product that you need to mix with water, it's extremely important to use non-chlorinated water when mixing. And then again, consider, is this formulation a liquid or is it a powder that you need to mix up? And then is this application a seed treatment, a drench, or a foliar application? All of these variabilities will inform the way that you use the product and the goals that you will have when identifying products to incorporate. And then finally, this inclusion of biostimulants needs to take place as you are practicing good soil health principles, right? So this will only increase the beneficial outcomes of practicing all of these other principles that are minimizing soil erosion and loss. We want to think about responsible and effective nutrient management. And then finally, the best use for these products is an integrated pest management, including different cultural practices, rotations, and considering all of the other strategies for minimizing disease. So with that, I'm happy to share my information, and please feel free to reach out if you have any particular questions about the things I talked about here or additional questions.
Awesome. Thank you, Mallory. So we'll open it up for questions. Um, Kind of one of the things that I was thinking about when you were presenting, you mentioned how, in specifically the field of biostimulants, we have seen a lot of products kind of on the market. And is there a resource that you would recommend or that you might know about in terms of like a database or a particular certification where folks might be more confident in the legitimacy of that product or the claims of that product being legit?
Yeah, unfortunately, the answer is no. So there is that limited voluntary certification program. As far as additional, trials, your local extension agent is the best, but I know it's this vast market. So I know that there are not extension run trials for all of these products. My team is building a database of field trials that we have installed at the moment. Most of these are on row crops, but we're moving into vegetable production this season. So I'm hoping to build a database. Um, I would say for testing yourself, right, there's important considerations, including controls and thinking about how you're installing the trials and trying to keep all of the other variables to a minimum. I know that can be challenging, right, when we're trying to rebuild and sort of improve production to kind of do a bunch of things. But in order to evaluate the efficacy of a single product, we really need controls and untreated. So I know that's not a satisfying answer, and hoping to build out that database.
No, yeah, no, that's great. That's the reality, right, that we live in. So I appreciate you speaking to that. Rachel, do you have any questions?
Excuse me. One question I had was, do you know if there's been research studies or how the microbiome changes after a flooding event? Whether diversity of the microbes or, yeah, what change occurs?
Hmm. The short answer is yes. Um, and there's so many variables and considerations. So often what happens after an extreme flooding event is um, if there is not movement of soil, that soil tends to go anaerobic, right? So the flooding kills off microbes that need oxygen. There are many microbes in the soil that don't need oxygen, right? So typically after a flood, that die off of microbes, microbes recover, right? So microbes that are anaerobic then begin to grow, and then other microbes establish. Typically, I would say on the recovery of on the scale of weeks. But what community is recovered will also depend on what was there before or if new soil was moved into a region. So I know a lot of the sedimentation issues, if it was other topsoil sedimentation, right? That's like similar type of agricultural microbes. If that sediment was something from a riverbed, certainly that'll be different microbes. From a wastewater treatment facility, different types of microbes. So it'll depend on how long the soil was waterlogged, if there was movement of soil, and then also conditions during that recovery phase. But there are typically, in the succession of microbial communities, there are known players, right, that are sort of like first to the scene after a soil is disturbed. And so that the succession of that community is somewhat well described for different disturbances.
And another question I had is, I'm familiar a lot with maybe the common biopesticides that people utilize, but maybe, or those even biostimulants that would have an application in for a pesticide use or whatnot, or disease control. But for those that are just biostimulants, in this case to improve soil health, are there common ones that people are using?
Yes. So, I think that the promising opportunity here is targeting that mode of action, right? So, in conjunction with soil fertility testing, there are microbes that solubilize phosphorus. So, if you have low phosphorus or, want to adopt approaches that are minimizing overuse of fertilizer, adding something like a phosphorus solubilizing microbe might be a good approach. There are other microbes that are nitrogen fixers, right? So in addition to adding legume cover crops, one could consider adding rhizobia in addition to cover crops or inclusion of legumes. For the most part, the inclusion of rhizobia in locations that have had previous legume production, there's little impact. However, following soil disturbance, if we're not really quite sure what's in our soils anymore, that might be a good approach to add a rhizobium into a cover crop mix that also has legumes. Let's see, other sort of biostimulants. There's a number that are generally plant growth promoting microbes, and then of course mycorrhizal. When considering vegetable production, right, brassica do not associate with AMF, so adding a mycorrhizal inoculant to your broccoli wouldn't be necessarily the smartest choice there, but there's some pretty compelling evidence about the inclusion of mycorrhizal for enhancing like tomato production and other vegetables.
Thank you.
Yeah, and that's all the questions I have. So thanks again, Mallory, for being able to present to us today. And if you have any questions after viewing this video, reach out to your local extension agent or contact Mallory, and we'll definitely try to answer some of those questions that you might have. But thanks again for talking with us today.
Thank you for the invitation.